Welcome to chapter 24-5. We're talking about the pancreas. So our pancreas, just like we talked about with complete anatomy, is going to be very in close proximity to the stomach. And it's going to have a very, let me see if I can hide this. It's going to have a really nice duct that will, a duct going right through the pancreas, collecting all of the enzymes that it's creating and dumping it directly into the duodenum. You can see that connection right there, dumping it straight into the duodenum. So we're going to talk about why the pancreas is important and what it's actually doing to help with digestion. All right, so if you remember, the pancreas has two features. It's endocrine functions, which we already talked about in chapter 18. These are the hormones, so think glucagon and insulin. Those are coming from the pancreatic islets. That's the 1% of the pancreas, et cetera, et cetera. Now we're going to talk about the 99% features of functionality of the pancreas. Those are the exocrine functions. These are the enzymes that are getting secreted straight into the duodenum. They're going through the pancreatic duct. The pancreatic duct meets up with the bile duct and blends, becoming the duodenal ampulla. So here's a cartoonistic picture of what's going on. So we have the pancreatic duct, I'll abbreviate PD, and then we have, let me actually draw it to what's actually going on. So here's the duodenum, here's our stomach, here's our pancreas. And if I were to, and here's our liver, and here's our gallbladder. And if I were to zoom in on what's going on right here, and you'll have to envision that the gallbladder is meeting up also at this junction. If I were to zoom in on that, I would have a duct coming from the pancreas, so pancreatic duct. And then I would have a duct coming from the gallbladder, called the bile duct. And those are going to blend together and create an ampulla, which is named or an ampulla is named after like a goblet shape. And this ampulla will dump directly into the duodenum. All right, and since this is dumping straight into the duodenum, we're going to call it the duodenal ampulla. And so we have a better picture of this coming up, but just in simple pictures, what does that look like? All right, the pancreas itself has three parts: a head, a body, a tail. Head being the most away portion that isn't close to the duodenum, and the tail being the closest portion to the duodenum. It's retroperitoneal, so remember this is similar to the kidneys where it's behind the peritoneum. This is why it was really difficult for a lot of you to actually find the pancreas until you really got in there and started feeling around and playing with the intestines in the cats. We have the pancreatic islets, and the pancreatic islets are what are doing the endocrine functions. Those are surrounded by pancreatic Acini cells, and those are what are making the enzymatic or the enzymes, and all of those cells congregate and dump their enzymes into the duct, the pancreatic duct, and then that shoots down to the duodenal ampulla. We call this pancreatic juice. So it's basically a very basic, watered down enzymatic juice that gets dumped into the duodenum. Here's some nice pictures of the duodenum wrapping around the pancreas and the pancreatic duct going deep down into the duodenum. And on the right-hand side, we have a cartoonistic picture of what's going on with pancreatic duct and those Acini cells making their enzymes and dumping it straight into secreting them into the duct, and then under the scopes what it looks like. But you're already familiar with what the pancreas looks like under the scopes. So once chyme leaves the stomach and actually enters into the duodenum, that will activate a hormone called secretin. And secretin will do a couple of things, one of which is tell the pancreas, hey, you need to start releasing those enzymes you've been making into the duodenum. We need them now. So secretin tells the pancreas to dump those enzymes into the duodenum, and this will make the currently acidic chyme become more basic or more neutral. because we don't want to, again, ruin the intestinal lining of the small intestine, and it's not built with the same structure as the stomach is, so it can't withstand that kind of pH. So we have to bring the pH back to a neutral pH or a more basic pH so that we can continue with absorption, or digestion, and then eventually absorption of those monomers. We're also going to have cholecystokinin. This is CCK. This is being released from eye cells in the nearby area. And those are also going to tell the pancreas to continue to secrete their enzymes. We'll have cranial nerve 10 coming in and helping, some branches of that, also telling the pancreas to promote secretions. And remember that these enzymes are being secreted. The enzymes that are being secreted are inactive. If they were activated, this would be really bad news for the pancreas because they would essentially destroy the tissues of the pancreas, and we don't want that. We want them to come out inactive, and then we can activate them in the duodenum and then start breaking down whatever it is that they need to break down. So what are the enzymes that are being secreted? We have pancreatic alpha amylase. This is helping break down carbohydrates. The other place where we have amylase is in the mouth. So we have salivary alpha amylase. That's the beginning portion of where we're breaking down carbohydrates. Then the second place we're breaking down carbohydrates is in the duodenum with the help of the pancreas. Then we also have pancreatic lipase being released from the pancreas. This is going to help break down lipids, and we'll talk a lot about how lipase is involved in that process. And then we have nucleases. Nucleases are going to break down DNA and RNA. And then we have proteases, and this is these are the big ones that come out in their inactive form. And we have trypsinogen, chymotrypsinogen, procarboxypeptidase, and proelastase. Don't worry about proelastase. Let's focus on the first three. So trypsinogen gets converted to trypsin, and trypsin is the active form. Trypsin can then convert chymotrypsinogen into chymotrypsin. And trypsin can also convert procarboxypeptidase into carboxypeptidase. Now, based off the names, you can kind of imagine what they're breaking down things into. So the carboxypeptidase, that's a peptidase, so it's breaking down peptides. Trypsin, that's also breaking down peptides. Like I said, don't worry about elastase. And then that's what we're working with. That plus the pepsin in the stomach, those are going to help us break proteins down. But other than that, we've got nucleases. Those are breaking down the DNA and RNA. We've got lipases. That's going to help us start the process of breaking down lipids. And then we've got amylases, and those are going to help us break down carbohydrates or secondary to what was going on in the mouth.